Hi, I'm Matt Fuxjager. I'm currently an assistant professor in the Department of Biology at Wake Forest University. I'm here today doing highlights for an article I recently published in Molecular Endocrinology called Hormones, Genes, and Athleticism, Effect of Androgens on the Avian Muscular Transcriptome. Now, this is work I did as a postdoc at UCLA with Dr. Barney Schlinger. In this project, what we do is we explore the way that androgens influence the transcriptomic profile of the skeletal muscles in two species of birds. One is the golden-collared manakin, the other is the zebra finch. In the golden-collared manakin, we know that males court females by performing a physically elaborate display in which they produce so-called wing snaps. Wing snaps are essentially when a male takes its wings and rapidly hits them together above its back to produce a loud firecracker-like sonation. Zebra finches don't produce this behavior. Instead, they court females by singing, and thus they use muscles in a vocal box called the syrinx. So what we did is we manipulated androgens in both of these species. We dissected the muscles out and used RNA-seq to measure the effect of that manipulation on the muscular transcriptome. We looked at this independently, but then we also compared these effects between the two species. And what we found were a number of very interesting things. Our paper goes through a lot of different analyses, so I'm just going to highlight some of the most important things that we found. First, we found that androgens influence the muscular transcriptome of the manakin much more than they do of the zebra finch. So, androgen treatment explains a lot more variation in the data we see than it does in zebra finches. And we think this is likely due to the fact that the manakin display itself is androgen dependent. And we have past evidence suggesting that this effect of androgens occurs in part, at least, through these skeletal muscles. So this kind of rounds out that nice picture for us and suggests that some of these effects occur because androgens are acting at the level of the muscle to modulate the transcriptional profile there, which in turn impacts how they can snap their wings together. Now, we did a GO-term analysis, and we found that a lot of the functional effects of androgens on the muscles through modulation of gene expression relate to things like calcium trafficking, ion shuttling, contractile machinery and contractile dynamics, among other things. We also identify a number of novel transcripts that are also related, we think, to muscle function. So we found a lot of these novel transcripts were myosin-like genes, suggesting that it's possible there's new myosin fibers in these muscles that are also important for the ability to produce this display. In our analyses, we are also able to pinpoint a number of manakin-specific androgen-dependent genes that we think are especially important for this extraordinary courtship behavior, these wing snaps. And a lot of these genes, interestingly enough, were related to lipid metabolism, suggesting that one way that androgens might prime the muscles so that manakins can produce their display is by enhancing or augmenting fuel efficiency in these skeletal muscle tissues. Now, of course, there is also a number of conserved effects of androgens on muscles between the two species, so we can't say that all the effects were unique. Another notable thing we found was that the effects of androgens on the muscles actually differed according to the actual muscle we were looking at. So androgens affect certain muscles in certain types of ways, but they affect other muscles in other types of ways. This level of diversity in terms of androgen-muscle interactions has not really been explored, and it may explain some of the kind of variation we see in the animal kingdom with respect to how androgens influence behavior by acting at the level of the skeletal muscle. In closing, I'd also like to say one of the benefits to our study is that we were able to provide, through our transcriptomic data, a richer analysis or a richer annotation of the manakin genome. And this is very exciting because there's a lot of people who are interested in avian genetics, avian genomics, and we hope that this will provide a nice resource to move forward in this field. So with that, I thank you for your time and I hope you enjoy the paper.